Welcome to week 4. We're actually going to take 2 weeks to cover this chapter, so this is for weeks 4 and 5. Chapter 4 covers neurophysiology, neurotransmitters, and the nervous system. So first we're going to talk about neurons. A neuron is basically just a nerve cell. You can see an example of one in figure 4-1. These are excitable cells. We'll talk more about how they're excited, that can receive and transmit information, and we'll talk more about that too. Just to step through the parts of a neuron, like all cells, it has a nucleus, that's where the DNA is. It has a membrane surrounding a cytoplasm. This is all what makes up the cell body, which is also called the soma. And then so all of that was common to all cells. Now, specific to neurons are dendrites, which are the fingers that receive messages from other neurons. And then the axon is the long process that extends out. This is what transmits the messages, and again, we'll discuss that in a bit. The axon hillock is where the axon attaches to the cell body. Then the myelin sheath is a layer of fat surrounding the axon, but you see the areas of the axon that are not covered by the myelin sheath are the nodes of Ranvier. And then at the very end of the axon are axon terminals. So we need to talk about the resting potential of the neuron. Resting potential is just the potential difference, or the difference in electrical charge across the membrane, that is from the inside to the outside of the membrane. This difference is about negative 70 millivolts. Millivolt is 1/1000th of a volt, and negative 70 millivolts just means that the inside of the cell has a greater negative charge. It's a more negative ions are on the inside of the cell. So ions, remember from chapter 1 that ions are molecules that have an electrical charge. These can be large drug molecules, or they can also be small molecules like potassium, sodium, and chloride, which are relevant for our discussion here. And then to create that resting potential across the neuron membrane, you can refer to figure 4-2, and there's several different processes that are needed to create this resting potential, both passive and active. First, the passive ones are diffusion. Recall from earlier, diffusion is just a substance moving from high concentration to low concentration. Electrostatic charge is another passive force where negative ions repel each other, positive ions repel each other. So a negative ion tends to want to be in a positive environment, whereas a positive ion wants to be where more negative ions are. The last passive process to consider is ion channels. These are just proteins that are in the membrane that allow ions to pass through the membrane. Importantly, these have differential permeability, which means not all ions can pass through equally. So we talk about potassium and chloride channels. They're non-gated, they're open all the time, but diffusion kind of evens out the force of electrostatic charge. So with the example of potassium, potassium is more concentrated on the inside of the cell, but the inside of the cell is also where more negative charge tends to be, so effectively these even out. Then there's an active process with ion pumps, and ion pumps are protein molecules in the membrane that selectively move ions from one side of the membrane to the other, and they can do this even against the concentration gradient of that ion. They require energy to do this. So the example here, the important ion pump to know about here is the sodium-potassium pump. It pumps 3 sodium ions out of the cell, and for that exchange 2 potassium ions into the cell, and that keeps an excess positive charge on the outside of the cell, which means there's an excess negative charge on the inside of the cell. So now from the resting potential, we can stimulate the axon, causing an action potential. First, just some vocabulary. Depolarization means that the charge, the difference in the electrical charge across that membrane is going to move towards 0 or become less negative. Hyperpolarization means it moves further away from 0 or becomes more negative. And then the threshold, we're defining at negative 55 millivolts. A threshold, you know what that means. It's just a point at which something happens. That's the point where the resting potential breaks down and the action potential can be fired. So let's go through the process described in figure 4-3. Basically, as a neuron becomes depolarized, it moves towards that threshold, and at the threshold of negative 55 millivolts or so, voltage-gated ion channels open. These are ion channels that are sensitive to voltage, and again, the threshold is where they open. The sodium voltage-gated sodium channels allow sodium ions to flow into the cell. That's due to the concentration gradient as well as the electrostatic charge. Remember, it's more negative on the inside of the cell, so the positively charged sodium ions flow in. As you can see in figure 4-3, as the membrane potential rises, it becomes less negative, it goes more towards zero. The potassium channels open, and the potassium ions begin to leave the cell because of their concentration gradient. At some point, the cell actually reverses charge. It's more positive on the inside of the cell, and the potential reaches as high as 40 millivolts, which is the point where the sodium channels close. So no more sodium ions can enter the cell. And now, as potassium continues to leave the cell, it causes the membrane potential to start going back towards the negative side. And it actually hyperpolarizes the cell. It returns the charge to be beyond negative 70 millivolts. At that point, the sodium-potassium pump can restore the resting potential, again, pumping sodium ions back out and potassium ions back in. And then the action potential moves along the membrane down the axon, because sodium ions diffuse laterally. When they come rushing into the cell, they also diffuse along the axon, and that depolarizes and opens the voltage-gated channels farther down. There's sodium channels at each of these nodes of Ranvier, and so as the sodium channels, as the sodium ions diffuse down the axon, the voltage-gated sodium channels farther and farther down the axon open up, and this causes the action potential to move down the membrane, down the axon. So the all or none law, it just states that action potentials are always the same intensity. They're either on or off. It's a toggle switch, basically. If an action potential is fired, then it's fired. To convey the strength of a stimulus, you do that through the rate of firing. That's the rate law, and that's shown in figure 4-4, where on the left, a weak stimulus stimulates action potentials to fire, but you can see while the stimulus is on, it stimulates it fires action potentials a few times. Maybe there's 10 in there, maybe there's 8, I don't know. Then on the right side, you see that with a strong stimulus, action potentials, there's a lot more of them. There's more, again, the action potentials are the same intensity, but they're just fired at a greater rate. So far we've talked about the action potential, which takes place along the axon. Now we're going to move on to the rest of the neuron dendrites in the cell body. And what happens here is they're called postsynaptic potentials. It's a similar idea where ion flow, depolarization of a membrane happens across a membrane, but these potentials are variable and location dependent, as opposed to action potentials where we just talked about they're always the same. You can have excitation where voltage-gated sodium channels open and sodium ions rush into the cell, depolarizing the cell. And then if the axon depolarizes past the threshold, you get an action potential down the axon from that. The opposite of that is inhibition, where instead of the sodium channels opening, you get voltage-gated potassium channels opening, and that causes potassium ions to rush out of the cell. It hyperpolarizes the cell. You can also have chloride channels open, letting chloride rush into the cell, again hyperpolarizing the cell. and hyperpolarizing the cell, taking the charge farther negative, again takes it farther away from that threshold, so it's harder to fire an action potential. And the idea with summation of excitation and inhibition is just that neurons are receiving this type of information all the time, and they sum it together, they integrate all that information. The sum of these determines whether and how often action potentials are fired. Temporal summation just means that the neuron is stimulated close in time. The neuron's integrating what it receives over time. Spatial summation, it means that it's integrating what the stimuli it receives over space. And this is described in figure 4-5. On top you have temporal summation, on the bottom you can see spatial summation. And then the idea, action potentials from sensory neurons, the idea here is just that neurons in your sensory organs can detect changes in your environment. So in your skin, you detect heat or pain or pressure. In your ears, you detect vibrations. In your eyes, you detect, etc. You understand. Those stimuli can cause depolarization and then action potentials. So that transmits information about the stimuli. So the synapse is where information is transferred between neurons. The synapse is actually a gap. You can look back at figure 4-1 to see a synapse where the axon from one cell is in close contact with dendrites of another cell. The axon from one cell can also be in close contact with the cell body from the next cell. And that's the synapse, that gap between the presynaptic cell and the postsynaptic cell. Now, synaptic vesicles are vesicles, think of little bubbles that contain neurotransmitters. These vesicles can fuse with the presynaptic cell's membrane, and that can dump the contents into the synapse. This is called exocytosis, and the contents are neurotransmitters again. So then the neurotransmitters diffuse across the synapse to the postsynaptic cell, and we'll discuss several different specific neurotransmitters soon. Neuromodulators are just that. They modulate the effect of neurotransmitters. They don't depolarize a cell in themselves, but they can increase or decrease the effect of neurotransmitters. Receptors are proteins in the membrane of the postsynaptic cell. You can think of them as receiving neurotransmitters, or they're where neurotransmitters dock, or where they bind, however you want to think of it. And these are specific for the specific types of neurotransmitters. Ionotropic receptors are receptors that are connected to a gated ion channel, so neurotransmitter binding of those receptors causes the ion channel to open or close, whatever the case may be, which then affects the flow of ions and can either hyperpolarize or depolarize a membrane. And then metabotropic receptors is where the neurotransmitter binding causes a cascade of effects in a cell. Now sometimes that could be, it could affect an ion channel nearby, but it can also be more indirect. It can activate secondary messengers. And these type of receptors are shown in figure 4-6, where you can see the G protein might go on to affect an ion channel nearby, but it can also activate secondary messengers. The most important secondary messengers are cyclic AMP, cyclic GMP, or a kinase. So cyclic AMP activates protein kinase A, cyclic GMP activates protein kinase G. Kinases are just enzymes that act on specific proteins. They add a phosphate group to proteins. And that little change can make a huge difference in whether a protein is activated or deactivated, how active it is. It can activate or deactivate ion channels, receptors, pumps, transcription factors, which then control whether specific genes are transcribed. So kinases, these secondary messengers and kinases affect all different kinds of processes, cellular processes. The book mentions this may even be how memories are formed. They can be a lot longer term than an action potential, which is taking place over less than a second. You can see figure 4-7 for an illustration of this, where secondary messengers at the membrane, this is all on the inside of the cell, can affect kinases, they can affect transcription factors, and you see the scale on the left is the time, the duration of the action, and again it goes from seconds to hours to permanence in quotes there. Transcription factors turn on gene expression or turn off gene expression, and it's not permanent, but it can be relatively permanent. So this is all, you don't have to know the specifics on this, but just know that these secondary messengers can have a lot bigger effects than just starting an action potential. So neurotransmitters can also affect the presynaptic cell. The book talks about autoreceptors, which auto just means self. These are metabotropic receptors that detect their own neurotransmitters. They regulate the level of neurotransmitters, and if they detect that too much is available, they'll synthesize less, and vice versa. If they detect too little is available, they'll tell the cell to synthesize more. Then heteroreceptors, hetero just means other. These are metabotropic receptors that detect chemicals from other nearby cells. Retrograde signaling is where information from the postsynaptic cell is transmitted to the presynaptic cell. This is how a postsynaptic cell can modulate the presynaptic cell, the amount of neurotransmitters synthesized or released or whatever the case may be. And this can be through either excitation or inhibition. Depolarization-induced suppression of excitation, depolarization-induced suppression of inhibition, depending on what exactly the effect of the retrograde signaling is. And then finally, at some point, you need to stop the action. You need to stop the transmission of neurotransmitters. You can do this either by reuptake, where the presynaptic cell reabsorbs the neurotransmitter and effectively recycles it, or deactivation, where an enzyme degrades the neurotransmitter and then again it can be recycled. The nervous system is made of the central nervous system and the peripheral nervous system. The central nervous system is the brain and spinal cord. We'll talk about that in a minute. And the peripheral nervous system is everything else. So that's what we're going to talk about here. Both the central nervous system and the peripheral nervous systems are organized where cell bodies are grouped together and axons are bundled together. And just a little vocabulary. In the central nervous system, those groups of cell bodies are called nuclei, and those bundles of axons are called tracts. In the peripheral nervous system, those groups of cell bodies are called ganglia, and the bundles of axons are called nerves. The peripheral nervous system, that is everything outside the brain and the spinal cord, is organized further into the somatic nervous system and the autonomic nervous system. So the somatic nervous system is the things that we're conscious of. So we have sensory nerves that receive information from the environment about heat or cold or pain, or you can hear or you can see or you can taste. Those are sensory nerves that are receiving that information. And then motor nerves are just what they sound like. They're controlling glands and striated muscles, the muscles that we control. The book also mentions cranial nerves that control basically your face and your neck. You can move your face around, and those are all things that you're conscious of. The other side of the peripheral nervous system is the autonomic nervous system, and this is concerned with things we're not usually aware of. The examples the book gives are blood pressure, your organ function, your hormone levels, things like that. And then again, it controls muscles, but it's, again, systems we don't consciously control, your heart, your intestines, muscles like that. And that's further divided. The autonomic nervous system is further divided into the parasympathetic and the sympathetic nervous system. These are separate systems where you can see in figure 4-8 the ganglia originate from different points of the spinal cord or the brain. They use different neurotransmitters. They're separate systems. The parasympathetic system is, the book calls it rest and digest. It's kind of the maintenance of your body, the day-to-day living that you need to do to stay alive. The primary neurotransmitter for that system is acetylcholine. And then the sympathetic nervous system is the fight or flight response, where you can respond to something that comes up suddenly. And epinephrine or adrenaline is the primary neurotransmitter there. And the point here that they have separate primary neurotransmitters, acetylcholine for the parasympathetic system and epinephrine for the sympathetic nervous system, since they use different neurotransmitters, drugs that affect acetylcholine or adrenaline will only affect one or the other of those two systems. So now we're moving on to the central nervous system, which is again the spinal cord and the brain. The spinal cord is primarily a relay station. Information from your sensory nerves are given to the brain via the spinal cord, and then motor commands are given from the brain to the motor nerves. You can see this in figure 4-9. You've heard about white and gray matter in the brain before. This is in the spinal cord too. The white comes from the myelin sheath around the axons, and then the gray is the unmyelinated parts of the nerves, the cell bodies. And then dorsal versus ventral, think of a dolphin's dorsal fin on its back. Ventral, I think of your lungs are how you breathe. So your lungs are in the front. So sensory nerves enter at the dorsal side, which is the back side, and motor nerves leave from the ventral side, which is the front side. Then your brain has an incredible number of neurons. 10 to the 11th is 100 billion. These all have thousands of connections. They all make thousands of connections to other neurons, thousands of synapses per neuron. Neurogenesis is creating new neurons, which you do throughout your life in specific parts of the brain, the hippocampus and the subventricular zone. We'll talk about those specific parts of the brain in a bit over the next few slides. And then neuroplasticity is not where neurogenesis is creating new neurons. Neuroplasticity is reorganizing the neurons, the connections of the neurons, and that happens due to learning and development. It can also repair some damage to the nervous system. You can see figure 4-10 has an overall look at the brain, and we're going to go into more detail on each of those structures over the next few slides. So for the next few slides, we're going to go through specific parts of the brain. We're going to go through a general introduction to the anatomy of the brain. For the purposes of this class, I'm not expecting you to know each of the structures in the next few slides. Please don't spend your time trying to memorize all of these different structures. You can refer to figure 4-10 for an overview of kind of a roadmap of where we're going. We're going to start with the hindbrain structures. Hind means back. The medulla oblongata is at the very base of the brain. It's controlling your autonomic nervous system, things like your heart rate, breathing, vomiting. The pons is just above the medulla, and this is involved with sleep. It's a bridge between the cortex and the cerebellum, and actually pons means bridge. The locus coeruleus, part of the pons is called the locus coeruleus, and it's involved with things like fear, anger, panic. It can detect novel stimuli, or it's involved with the detection of novel stimuli, which makes sense. That's why we're often, we're often afraid or panicking because of something new, and that's because of that structure. The cerebellum is in the very back of the brain, and it's involved primarily with motor control, motor refinement. And then moving to the midbrain structures, the reticular formation, there's both descending, the descending reticular formation going down to the spinal cord. That's controlling the autonomic nervous system, your reflexes, things like that. And then the ascending reticular formation is also called the reticular activating system. And it's going up to the cortex of the brain, and it's involved with attention and wakefulness. Periaqueductal gray area of the brain is involved with pain and defensive behavior. There's also a punishment system there. The substantia nigra we'll talk about more in a little bit. It's involved with motor control. The ventral tegmental area has a reward circuit, and this one is a good one to know, or for you to remember because of its role in addiction. Again, not rewards, the reward circuit is how drugs can be strong reinforcers. So all of these structures are important, but again, please don't memorize them. One important point here is that all these different structures have different neurotransmitters as their primary ways that they convey information. And because of that, different drugs have different effects on different parts of the brain. So the one structure that I mentioned on the last slide that is important to know, to understand, is the VTA, the ventral tegmental area. This is important to understand because of its role in addiction. And we'll talk about this more in chapter 5, but I just wanted to spend a slide on it here, just highlighting it. It's part of the mesolimbic dopamine system, which is the reward circuit in your brain. That reward system works for both natural reinforcers and drug reinforcers. There's neurons in the VTA that produce dopamine that then project to a bunch of different areas in the brain. And the book lists a bunch of them, but I want to highlight the nucleus accumbens. And you see that in this image here, the green line, it's supposed to represent neurons going from the VTA to the nucleus accumbens. The nucleus accumbens normally inhibits motor and movement systems in your brain, but when the VTA floods dopamine into the nucleus accumbens, that inhibition is inhibited, so motor functions are activated. So next, let's talk about the structures in the forebrain. The basal ganglia just means ganglia is a group of neurons. Basal means it's at the base. It's at the bottom of the cortex, right underneath the cortex. And this is involved with movement and cognition. The book talks about Parkinson's disease. This results from too little dopamine, and that obviously causes motor effects, and you can treat it with dopa, which is a precursor of dopamine, basically just flooding in the dopamine that the brain is missing. The limbic system is hard to define exactly. The book calls it an ever-expanding megasystem, and that's because the definition's evolving and it's very highly interconnected. This system is involved with learning, memory, emotion, planning, kind of complex functions like that. And the book makes an interesting point where it makes sense that it's so highly interconnected with all these different parts of the brain are so highly interconnected because you need emotion to make memories. It's hard to separate memory from emotion. You need motivation to learn. You can see figure 4-11 for some of the components of the limbic system. We already talked about the amygdala. This is responsible for fear and anxiety, aggression in animals. Patients with post-traumatic stress disorder have hyperactive amygdala. Then the hippocampus is another structure to note. This is involved with learning and memory. A long time ago, it used to be removed in patients who are having severe seizures or epilepsy, and it caused amnesia. Rats who have had their hippocampus removed also have trouble with simple spatial memory, completing mazes and that type of thing. The thalamus is a relay center. It relays sensory information to the cerebral cortex, and it also relays between the cerebellum and the cerebral cortex. Again, the cerebellum controls movement. Then the hypothalamus is a tiny part of the brain. The book describes it as the size of a pearl that regulates homeostasis, so that's things like your body temperature, your blood pressure. It also regulates metabolism, so it's going to tell you if you're hungry or thirsty, and it maintains your circadian rhythms. It does all those things by acting through the autonomic nervous system, the descending reticular formation going down towards the spinal cord where the autonomic nervous system is controlled. And it can also do that via your pituitary gland by controlling the hormone levels in your body. Then the cortex is the surface of the brain. This is what you think of when you picture a brain. It's about 3 millimeters thick.
It integrates sensory information to bring understanding, what we call understanding. You can see a lot of these areas in Figure 4-12. And a lot of them just make sense. The visual cortex integrates images. This is located at the occipital lobes at the back of the brain, or at the back of the cortex, sorry. The auditory cortex is responsible for integrating sound. That's in the temporal lobes. Somatosensory cortex is where we sense taste and sensory information from your body. That's in the frontal lobes. Motor cortex commands motion, again in the frontal lobes. The prefrontal cortex is in the very front, and this is where higher mental processes happen. Things like abstract thoughts and decision making, planning, and judgment. Two last areas to mention: cingulate cortex and the entorhinal cortex are part of the limbic system. They're involved in attention, craving, learning, and memory, so they have implications for drug use. The developing nervous system in a fetus is very susceptible to disruption. It's very delicate, we'll say. So if you can imagine normal cell development, it takes very specific signaling at specific times and in specific places for cells to form, for cells to be differentiated. That just means to become nerve cells or heart cells or muscle cells or skin cells, whatever. For cells to migrate to where they need to go. For a neuron, axons need to be projected and then need to form synapses with other neurons. And so all of that takes specific signaling. It's very delicate, and any chemicals that the mother ingests can disrupt the proper development of the fetus. So teratogens are any drug that cause the normal development of a fetus to be disrupted. Thalidomide is one example of this. It was often given in the 60s for mothers who were suffering from morning sickness. It helped their nausea. Side effect was that babies were often born with missing or malformed limbs. And behavioral drugs can also cause these types of effects. One example is alcohol. We don't know the exact level that's safe to drink, but I'm sure you've already been aware that fetal alcohol syndrome can result if the mother drinks while pregnant. And then functional or behavioral teratology. The idea here is that there's other drugs that have more subtle effects on a fetus that can cause problems. So recall from a little bit ago that neurotransmitters are what allow a signal to be passed from neuron to neuron. After one neuron has fired an action potential down its axon, this is the way that the signal can pass beyond the synapse to the postsynaptic neuron. Page 79 has criteria to define a neurotransmitter, but I think this figure 4-13 is a good way to understand the steps in neurotransmission. So up at the top, number 1 is neurotransmitter molecules are synthesized from precursors under the influences of enzymes. So that's taking the purple quarter circles to form purple circles. The enzymes are the springy looking things, and it's just synthesizing those precursors. You don't have to know specifics on how this happens to form the purple circles, the neurotransmitters. And number 2, neurotransmitter molecules are stored in vesicles. We had talked about these vesicles a few slides ago, and you see these neurotransmitters grouped together inside this circle. Number 3, if neurotransmitters leak from their vesicles, they're destroyed by enzymes. But then number 4 is the more interesting part. Action potential causes the vesicles to fuse with the membrane, and that's what dumps the neurotransmitters into the synapse. I think the word we used a couple slides ago was the cell exocytoses the neurotransmitters. So then once the neurotransmitters are in the synapse, either number 5, they can bind with autoreceptors, and that's how
the presynaptic cell can sense whether there's enough of that neurotransmitter available. If the autoreceptors bind a lot of those neurotransmitters, the subsequent neurotransmitters will be inhibited, whether its formation will be inhibited or release will be inhibited. Number 6 is where the signal is actually passing. The released neurotransmitter molecules bind to postsynaptic receptors. We talked about that, what they do, a few slides ago, whether those receptors are directly connected to ion channels that help depolarize or hyperpolarize an axon, or whether they're more complicated and go through secondary messengers or that type of thing. And then number 7, the released neurotransmitter molecules go through either reuptake or degradation to get rid of them. So drugs can act as agonists or antagonists towards neurotransmitters. This is figure, 4-14 in your textbook, Mechanisms of Agonistic and Antagonistic Drug Effects. On the left side, you've got a bunch of different agonistic actions that drugs can take, and on the right side, antagonistic. So the difference between agonist and antagonist means agonists facilitate communication of a particular neurotransmitter. They help something happen. Antagonists impede that communication, so they stop a neurotransmitter from exerting its effect. Now there's different ways that this can happen. Direct agonists direct agonists bind a receptor. They mimic the neurotransmitter. So it would be, the second to the bottom on the left side, drug binds to postsynaptic receptors and either activates them or increases the effect on them of the neurotransmitter. That's a direct agonist. A partial agonist binds the receptor and only activates it weakly. Then a mixed agonist is an antagonist for one receptor type but an agonist for another type. That's mixed agonist-antagonist drug. Direct antagonists bind receptors and keep neurotransmitters from binding. So that would be the bottom, on the bottom of the right side, a drug. This is also called receptor blockers, direct antagonists. So it's a receptor blocker. It binds to the postsynaptic receptors and blocks the effect of the neurotransmitter. And that can also happen with autoreceptors. The second to the bottom on the right side, drug activates autoreceptors, inhibiting neurotransmitter release. So indirect agonists, indirect antagonists don't mimic the neurotransmitter, but they just affect the receptor in other ways. Non-competitive binding is one example that the book gives of this, but there's a bunch of other examples right here on this figure where maybe a drug increases the release of neurotransmitter molecules, or a drug, an indirect antagonist might be that the drug could block the release of the vesicles, something along those lines. So there's agonists, antagonists, and direct as well as indirect that you should be aware of. There are a lot of different types of neurotransmitters. Most are small molecules, but you can also have peptides. They're called neuropeptides or neuroactive peptides that are relatively large molecules. Many of these are more commonly known as hormones, but common neurotransmitters of all types are listed in Table 4-1, and in this slide we're just going to really quickly look at types of neurotransmitters, and then we'll talk more about them as we go through the next few pages of your textbook in the next few slides. Acetylcholine was the first discovered. There's several different biogenic amines, monoamines. You know epinephrine and norepinephrine better as adrenaline and noradrenaline. Dopamine is also a catecholamine. Serotonin, you're familiar with. Amino acids are both excitatory and inhibitory. There's glutamate and aspartate. There's GABA and glycine. And then there's a bunch of different peptides. Peptides are basically just short chains of amino acids. And opioid peptides are morphine-like molecules. There's a lot of different things that we typically think of as hormones. Things like the TRH, the CRH, TSH, all the different hormones listed on the right side of that table 4-1. Insulin is a miscellaneous peptide listed down at the bottom. And again, there's a fuzzy line between hormones and neurotransmitters. The book mentions that they're sometimes called neurohormones. Just know that neurons are classified according to the main neurotransmitter they produce. So cholinergic neurons are producing and releasing acetylcholine. Adrenergic neurons are producing adrenaline or epinephrine. Dopaminergic neurons are producing dopamine, and so on. So this information is summarized in table 4-2, and I just want to spend a little bit of time going through some of the types of neurotransmitters in a little bit more detail. Acetylcholine was the first neurotransmitter discovered. Acetylcholine receptors can be classified according to which drugs affect them. So nicotinic receptors are stimulated by nicotine. Muscarinic receptors are stimulated by muscarine. Botox also is it blocks release of acetylcholine. So then, if that's in place, you can't move your facial muscles. The acetylcholine can't reach the next neuron. And that's how Botox works. You can't move your facial muscles to express yourself or sweat in your armpits or whatever it is, wherever the Botox is injected. Biogenic amines or monoamines, they're named that because they're synthesized from a single amino acid. Catecholamines are synthesized from tyrosine. You can see that illustrated in figure 4-15. The catecholamines include epinephrine, norepinephrine, and dopamine. Then serotonin is also a monoamine. It's synthesized from tryptophan. A lot of antidepressants target the reuptake and degradation of these molecules, making them more available. Cocaine and Ritalin also block the reuptake of them, making them more available again. And then dopamine, you've already heard me talk about, and we'll talk about it more. It's involved with, it's linked to Parkinson's disease because of its role in the substantia nigra. It's also involved with mesocortical, mesolimbic pathways, and that has implications for addiction in general, with motivation and rewards. The two amino acids I want to mention are glutamate and GABA. Glutamate is the most commonly used excitatory neurotransmitter in the brain, and GABA is the most commonly used inhibitory neurotransmitter in the brain. And because they're so commonly used, both of those types of neurons are widespread throughout the brain. They're not constricted to any one area, and both have several different subtypes of receptors. Adenosine, the book mentions. This is a major molecule that your cells use for energy with ATP and ADP. Secondary messaging, we've already talked about CAMP. It has a role in sleep and wakefulness, and its receptor can be blocked by caffeine to stop adenosine from binding. Caffeine blocks that receptor site, so you can't fall asleep. Endocannabinoids are small lipids. They're related to THC. Endo means they're inside of you, and cannabinoids, you know what cannabis is. So these bind to cannabinoid receptors. And then a bunch of different peptides. Opioid peptides are morphine-like molecules. The others, a bunch of others, are named for where they're made. For example, the pituitary peptides are hormones released from the pituitary gland. And then the other group is miscellaneous for everything that doesn't fit into other groups. There are a few different techniques that can be used to image the effect of drugs on your brain. First, we're going to talk about positron emission techniques. So positron emission tomography, or PET.
A patient ingests a radio tracer, so that's a molecule of some sort, whether it's a drug or glucose or water, whatever the case may be, a tracer labeled with a radioisotope. And then once that radioisotope reaches its half-life or starts to decay according to its half-life, then you can put that person in a scanner. As the radioisotopes decay and emit positrons, the positrons will create a gamma ray that can be detected by basically a ring of crystals. These crystals fluoresce when the gamma rays hit them, and then a computer is used to monitor the fluorescence. This technique can be used for a wide range of drugs. It's used to measure brain distribution and the activity of the drugs. The local concentration of drug receptors can be determined. You can use tiny amounts of the drugs that aren't metabolically active. They're not going to have an effect on the person ingesting the drugs, and still see what happens to the drugs. You can also assess competition for receptor binding or monitor brain activity by monitoring glucose metabolism. There are several problems with this technique. The spatial resolution's relatively low. It's a few millimeters, 4 or 5 millimeters, which makes it hard to determine specific sites within the brain where something's happening. It also submits a person to high levels of radiation, which obviously has some health concerns, especially if one individual will be measured multiple times. And then it costs a lot too. So there's few PET scanners, so that makes it hard to access. Some people don't have them anywhere near them. A related technique, single photon emission computed tomography, or SPECT. It's a similar technology to PET, but the radioisotopes used take a lot longer to decay. Their half-lives are on the order of hours or days. And so that makes it somewhat cheaper, because the radioisotopes can be created and then used the next day or whatever. It also makes it, not use the next day, but just you can monitor the person over the next day. It's not nearly as time sensitive of a process. You're also able to use this technique to monitor longer term processes, but its spatial resolution is actually even worse than PET. And again, it's going to submit a person to high levels of radiation. And even though it's somewhat cheaper than PET, it's still not inexpensive. Magnetic resonance imaging requires a magnetic field. A powerful magnet in an MRI machine is turned on, and the hydrogen in your body, protons in your body, will align with a magnetic field. And then radio frequency waves pulse, which causes the hydrogen it transfers energy to the hydrogen, the protons. When the pulse is turned off, protons release that energy, and that's what's detected. And basically, you can read the physics description in the book of how MRI works, but there's relaxation times for what happens to these hydrogen, what happens to the protons when the pulse is removed, and the relaxation times are different for protons in different environments. So that's how you get an image where different parts of your brain are different colors, or the cerebrospinal fluid is a different color. Functional magnetic resonance imaging, or fMRI, you inject a magnetic agent in the contrast technique. That increases the magnetic field where the brain is activated, so it generates an image of active as compared to inactive areas of the brain. Or, rather than injecting a magnetic agent, you can use the magnetic properties of iron-rich deoxygenated hemoglobin. This is called the non-contrast technique. Most common here is the blood oxygen level dependent imaging, or BOLD. Basically, when the brain is active, it consumes more oxygen, and that changes the ratio of deoxygenated to oxygenated hemoglobin. MRI techniques have higher contrast resolution and spatial resolution as compared to the positron emission techniques. They're relatively fast, they're non-invasive, they're not using radiation like we were with PET and SPECT. Patients still need to keep still for a duration of time in a relatively small space, so that can be a problem. And there's still some other issues with these techniques. So both PET inspect and fMRI and MRI, both types of techniques have their pros and cons. You can see a summary of those in Table 4-3, and you can also see examples of images produced by these different techniques in color plates B, C, and D.